I'm a genomics Aotearoa postdoc based at Manaaki, Whenua, and I'm looking at identifying genomic signatures of invasiveness for biosecurity. I'm the other half of the postdoc project that Ellie's working on as well. And so my part of this project is looking at the comparison of whole genomes of invasive and non-invasive species with the goal to identify traits that are associated with invasions. This is really with an end goal of hopefully developing an analysis pipeline and potential machine learning tool to predict the risk of unknown species to Aotearoa New Zealand in the future. So I'm going to kind of breeze through my background since I think I'll just be a bit of an echo chamber. We've had a lot of it the last 2 days. So yeah, I just have a few slides talking about New Zealand. So New Zealand actually has the second highest number of invasive alien species globally, only second to the USA, which considering our size is pretty spectacular. And in 2019 to 2020, yeah, I think it was 2.9% of GDP was the economic cost of pests, which was about 9.2 billion, which is an extremely high amount. So risk is escalating with increased travel and movement and with climate change, and our ability to detect and prevent invasions is currently quite limited. So biosecurity in New Zealand relies on two main factors of identification for pathways. We have the environmental factors, which is looking at climate matching models such as Climax to compare locations of similarly matched climates or meteorological variables to narrow the range of predictions to where an invasive species might emerge. And then we also have looking at risk pathways, which predict the biotic invasions based on identification of species that already have a record of invasiveness elsewhere. So this is just on the bottom here, an example of MPI's risk assessment process. They first focus on identifying a risk. Is it already on a list of pest species or a disease of concern? Is it present here already, and can it establish? And after this is decided on, it goes into a risk-based assessment. What is the likelihood of the establishment, the likelihood of then spread, and what are the overall impacts, either socially or economically? The four key points in this pathway rely on an organism actually having the mechanistic ability to invade. So what do we mean by that? When we talk about a mechanistic ability to invade, it's about the genomic architecture and the traits that are required by an organism to prevail. So we're looking at, especially in this instance, at gene duplication, SNPs, acquired traits that have environmental survival benefits and impacts. Duplications are an important source of novel phenotypes and are identified as a prominent feature of evolution. Increased gene copy number allows for increased plasticity and increased opportunity for adaptation to new environments. These sorts of selections are often associated with the lag phase of an invasion. It allows for a population to build up a preferential phenotypes, be that nutritional, defense-based, or metabolism. So in this project, the aim is to initially utilize different, I've called them levels of genomic data, to try and identify potential signatures of invasive species in known invasive species by literature. So we're using initially whole genome sequences to look at the genome architecture as a whole complete sort of basis. Other duplications, gene expansions and contractions, and then incorporating in Ellie's data as well, we're going to be looking at SNPs. We can move further down from the gene level into specific base pair changes that can alter gene expression and predict protein prediction, and identifying potential candidate genes that then refine our target based on whole genome comparisons. And as well as this, we're hoping eventually to have a look at microbiome accessory data sets, which we would like to include in our analysis. As Lee was saying this morning, microbiome is linked to fitness of a host, contributing beneficial metabolites, so it's very important to be included in these kinds of analyses as well. So what is our approach to this overall? It's sort of a two-pronged approach, with Ellie taking one side and myself the other. So for me, I'm looking at sifting through different data sets. So it's using GenBank, the I5K sequencing project, IUCN, and pretty much going through as much literature as I could feasibly find, looking for whole genomes of known invasive species, and those that are described as non-invasive. I've kind of shifted that recently because finding anything that is described as non-invasive is impossible. So I've now expanded that to something that's having a native range and expanded range, so I can kind of broaden it and have more sequences to include in my analysis. So then I've implemented phylogenomic analysis to look for changes in gene expansion and contraction. Ellie's been analysing the SNP data set of important pest species to build a candidate gene list from outlier SNPs, and then this data will be used to filter through author groups that I've identified through my phylogenomic analysis by protein, family, and function. The overarching goal of this is to kind of create a comprehensive feature list, which would start with initially evolutionary gene expansion and contraction. And by generating this, the idea is to feed this into a machine learning tool that we can hopefully accurately predict an organism's ability to invade based on these signatures. We're kind of limited at the moment to data sets which represent both invasive and non-invasive species within the population. So at this point in time, I'm looking at Diptera, Lepidoptera, and Hemiptera, and a combined approach that includes all three of these orders, as well as Coleoptera and Hymenoptera as well. Yes, so for this project, Alice went over BMSB, and she had a poster as well on Queensland Fruit Fly about predicting these candidate genes, and she came up with these great gene candidate lists of outlier genes that potentially have sort of adaptive function. Looking between the two data sets, we find that there's some things in common, specifically sodium channel proteins and odorant receptors, which I did note as well that some Deeptha found in her fall armyworm yesterday as well, so I've made a careful note of that. So these all share a candidate gene. So for me, this was a pretty good starting point to look at because there was evidence that it was shared across multiple species. And this is just an example of how I utilize these candidate genes. So for each of the sequences, I generated PFAM and Panther signatures using Interpro, where I just did masses of blasts to try and find out the best gene signatures to use for my analysis. So I then annotated the orthologous families that I found in my phylogenomic analysis as well, and filtered these based on the signatures that I identified from the various databases. So initially I started with the sodium channel proteins and the odorant receptors, and I couldn't resist the GST as well, since it was particularly important in insecticide resistance. I'll talk a bit about why these were selected and narrowed down from full genome data, but first I'm just going to show an example of some preliminary data that came from this that I only got last week actually. So it's just an example of the results of some gene expansion and contraction between the Diptera species. I included 8 invasive and 2 non-invasive species. The values that I generated were pretty large, so I just transformed these using log10 to standardize them and reduce the disparity before using colors to make them look like a heat map as well. Unfortunately, I didn't find any major trends observed between invasive and non-invasive, but I made myself feel a little bit better because I don't know if this actually works. But no, maybe not. But between Drosophila itself, looking at the invasive and non-invasive, although there's only 3 representatives, there is some trends in the sodium ion channels. So that gives me some slight hopes that there are some things that we can look at in the future as well. I'm not particularly surprised that there wasn't so much of a trend coming out. We've had a lot of talks this week that have talked about the limitations of data sets, and we're talking about how whole genomes are underrepresented in invasive species. Paige was saying 55% have a reference genome, and I believe this is a lot lower as well for non-invasive species. So it's been quite a challenge to be able to get the data sets to run these analyses. But this is, however, an example of the traits input I mentioned for the machine learning software, which we've tested on much larger data sets with varying results. Hopefully with further work, we can refine the traits list and identify more candidates or different analysis to try and find different types of candidates to include in these data sets as well, and to find more different kinds of patterns to include in the genomic data. I'm just going to run quickly over the premise behind the machine learning portion of the project. So MPI have, I think, thousands of incursions of unwanted organisms every day or every year, and to go manually through these and try and find any trait would be a mammoth task for anybody, never mind a team of people. So the idea is, alongside Tony Smith here at Waikato Uni, we're using a machine learning algorithm, specifically Weka, to try and build sort of a predictive tool that, based on the genomic signatures we find, would be able to allocate species into a highly invasive or non-invasive group category based on these selected features. So this would be using the filter traits from Ellie's work, and using a decision tree, the machine learning algorithm can then make its own decision in partitioning the data sets into different attributes of non-invasive, invasive. I've got an example of a decision tree I ran on Friday on here, and looking at it, it looks quite nice. It's very nicely split the data into invasive and non-invasive, which is potentially quite nice until you look at the numbers and realize that there's 7 invasive and 3 non-invasive, and that's my data is 70 to 80% invasive species. So really the margin of error is quite low. All it's doing is really predicting invasive species, which for me is another piece of evidence that non-invasive species are severely underrepresented in the data set, and we need to kind of increase sequencing efforts and sequence everything everywhere all the time. So that would be great for me. Yeah, so that is mainly the conclusions of where I'm at so far. Is moving past the individual case studies is quite challenging. It's challenging itself to look at individual population genomic databases. We've heard from people about how cost prohibitive it is to do whole genome resequencing, and how much better you can have it with that as well. And that's particularly the case in one species, but when you're trying to look across a whole order of insects, it's even more of a challenge. There's another reference to Paige's work here as well, about how there's only 55% of invasive species that are studied have a reference genome. For my project particularly, this is a bit of a problem, especially when we're trying to look at invasive compared with non-invasive, and you look at the metadata on particularly GenBack's really bad for it, is they're often just laboratory strains, or they don't say where they're from specifically or when. So as much as you can have a genome for Drosophila suzuki'i, and we know that's an invasive species, we actually don't know whether or not the origin of that strain itself is from the native or the invasive range, which I believe is quite problematic for especially what I'm looking at, because it would be nice to be able to have representatives of non-invasive and invasive. So I could ramble about that forever. So I think the databases are a problem, but I think it's something we can improve, and I think that's going to be a good topic for discussion for tomorrow. So yeah, my general conclusion of this is that the databases are a limitation, but we seem to be finding some trends, and I think it's heading in the right direction as well. I'm also inviting everybody here for an open discussion. We've had some really nice talks, and I've took lots of notes because everybody's had some really cool ideas, and I think if anybody has anything they might be useful, or any programs they've used that they reckon that I could get some benefit out of trying, please find me afterwards. I would greatly appreciate it. And as well as that, I would like to thank all of these people on this project, so Manpreet and Ange, Ellie and Tony, and invite any questions if anybody has any.
Cool, thank you, Amy, that was awesome. Does anybody have any questions?
Yeah, thank you. That is a really ambitious project. It's really cool. I'm wondering, and I know you're really restricted in the availability of genomes, but if you've thought about when you are in a space to be working with a larger amount of data, how to correct for phylogeny, and what you might see that's different just because of a closer shared history between species, versus actually being invasive or non-invasive, especially given that you're, as you said, the coverage of and availability of the data are already not perhaps what you would choose if you were able to do that.
I think this is a major thing, because the more you think about it, the more of an extent of data you can always want. The more I think about it, the more I'd want regional differences, and I'd want geographic or time dependent, like we were talking earlier. So I think this is also a problem. If I only have one representation of each species, then you're not really resolving the phylogeny. You don't know where it accurately sits. I think for this sort of project, it needs, I think it's going to need a lot of data.
Yeah. Well, good. Hopefully, as you said, we will sequence
everything.
Lots of things in the near future, and you'll have it.
Yes.
Hi, great talk. I was just wondering how you interpret insecticide resistance versus, or insecticide resistance genes versus invasiveness genes. I almost asked Ellie this earlier, but do you think of insecticide resistance as being an invasive trait?
I would say so. I would say that insecticide resistance is an invasive trait as much as anything else. Yeah, because it's a defense mechanism operating as well.
And we've just got one on Zoom as well that says, in Bacticera, Raphael found a small number of transcription, sorry, I'm so blind, differences between invasive and non-invasive sister species. Have you considered mining transcriptomic data?
Yes, there's a brilliant article I read not long ago that does exactly this with Drosophila and looking at the entire genus, and found that I think that it's a gene that's present in all Drosophila species that's upregulated in Drosophila suzuki that they reckon is what part of the function of their behavior in utilizing full fruit rather than rotten fruit to lay eggs. So yeah, I think it's a really important data set that would be great to use, but it's also another cost prohibitive one, and there's definitely not as much transcriptome data flying around as there is genomic. But yeah, there's some really good research on it.
Nice job, Amy. It's not really a question, it's more of a comment for the room as well, that we are currently looking for a third species to look at. So if anybody has any ideas, we are trying to focus on insects really as our first pass, but that's something part of the open discussion.
That was part of it, yes. If you've got any species that you think could be cool to look at.
Yeah, and we do need existing data.
Yes.
So.